So I have seen a lot of people are recently talking about this publication, including the Luciano Martellotto. He is a good friend. So the question was that with this AI revolution, artificial intelligence revolution, do you really need bioinformaticians or other similar people? And this thoughtful perspective by Marisa and Wilson, what they have tried to implement in here is that with AI integrations, you can do the bioinformatician will be able to do the AI design, complex discovery, and responsible work. But because AI cannot judge biological meaning or verify scientific validity, bioinformaticians remain essential. So I agree at some extent, and I want to go put a trial and by using one of my software that's been quite popular across the Cloud team. They have added these skills. This is the software that I have written. That's the universal biomedical skills and agents adapter connected with these, and here I have added up the skills, every single thing that's relevant to biomedical research and clinical work. For example, if you go to the genomics, you have all these that's you required for the genomics. If you go to the any other of them, for example, let's go multiomics, for example. So you have all the multiomics and everything in here that could be present in the market. Simultaneously, any part of it, it's integrated to agentic AI, and how it works, I will give you a demo. But for better understanding, let me just go through one of them. For example, this is genomics. Then we focus on single cell. So let's go to the single cell. Then we want to see, okay, how do you do the cell type annotation or something like that. I'm just giving you one example. It has every single thing that you required for any sort of the work within this. So you have this, and then you can go in here, you see that all the code that's required for the analysis is integrated. So in total, I think right now I have around 1800 skills. Those is integrated in here. Those are integrated in here, and oh sorry, 1207. I think it's not updated in writing. Total 1800 skills that's required entire into the biomedical and the clinical research and practice. Every single thing is in here, including AIME and everything. So I will show a demo. So first, let me just go through and take it out. Oh, so if you want to download it from GitHub, that's fine. If you want to install it from the pip, you can just take this part and get it done entirely. Just copy it and install it in Jupyter Notebook. Anywhere it will run functionally good. So let me just give you one example to prove these points. So let's say, okay, I want to download not all of them, just two groups. I downloaded this data set in here, and this is from this publication, right? So this is the publication from here. So I downloaded it. Let me just go to this. So this is the data set. Extracted it. Okay, so this is the tar files. Extract them all. And then we have this, right? .tsv file, tsv file .mtx files. This should be good enough for doing this analysis. Let's delete this. Now this is the set of skills we have in here. So you can just take any of the AI agents from here. So let us go through this. So this is the folder. Copy this. Let's say we take it from here. You can connect with any of the AI agents. I am not worried about it. You can connect with Claude, ChatGPT, and Gemini, DeepSeek, any of them. It doesn't matter. How it works, I will explain. Let's take the Claude. Just I'm giving you one example, not necessarily you need to take it. So we will simply write it down that what it need to be done. What is this paper about? What does it say? Okay, they are saying the B lymphoid cells are CD30 positive B lymphoid progenitors. Hemoglobin is then got more affected. Let's see. Write simple comment. So you say this is the manuscript. Yes. So I think for that purpose, it's not wise to download a couple of them. It's probably better to download all of this files in here. So let me just download all of them in there. So what I virtually did in here is that It had all the skills that's required, all the files that's required for analysis, and it's free to take these files and do the analysis. Plots, graphs to results. Correct. So I ask it to go through this and then read this manuscript and find the skills from this folder, take those skills and then analyze and prove that point. So how it will be working on that is that simply the skills I have built up in here, you can see it has universal biomedical skill and agent. So it has it connects with it works like a biomedical operating system. It connects with these skills, so which and then it goes into the agentic mode and run all of it one by one. So I'm just giving approval, and let's wait for a couple of minutes to get it done. So now let's go through the paper again. What does it say? Single cell analysis of bone marrow derived CD34 from the children with sickle cell disease and thalassemia. Let's mark it. So this is the one. Let us see what it does it say. In the conclusion, see, we have shown that compared with healthy adults, a high percentage of CD34 positive cells in bone marrow harvest taken from children with SCD and thalassemia are B lymphoid progenitor cells. Okay. The second part is that it reduces CD34 compartment. So if I look in here, and we look at B cells compartment size, in control lymphoid progenitor, it's 8.6%, here is 49 and 52%. Now see, this analysis is done, and how was the analysis done and how does it look? So let's go to the figures. These are checkpoints, all the .h580 file filtered, clustered, annotated by a lineage. Then in the figures, you can see you have the figure for genes per cell, UMI counts per cell, mitochondrial percentage before filtering, QC platter. When you have integrated them, you can see this is control thalassemia patient and sickle cell disease patient, and this is beautifully done. The author have done with the Rosura system, we are doing with the Python system, so there will be some variability. You can see the latent cluster, total 20 cluster, conditioned. I can see all this, integrated, lymphoid AU cell score. What was the lymphoid AU cell score marker? So if we go in here, so you can look at in here, cell identified B lymphoid project expression including CD17 and CD17B PARP, BPREV1, EVF1. So we use the same marker in here and to identify these specific lymphoid progenitors, and you can see the lymphoid cell by condition. So it's in control. This is thalassemia condition. This is sickle cell disease condition, and it's significantly increased up to 3 to 6, 8-fold. I mean, this is a huge difference. It's a kind of a synchronized with this percentage from 8% to 50%. So if we see in here, it's like around pretty close by 50%, 70%, same output. You can see in here the lymphoid progenitors in here, then the specific markers, all things so that you require for that. Now I think this is important. We are looking for the lymphoid progenitors. So you can see that this is control, and then in the thalassemia you have approximately 8%. You can see. So the percentage in here was by the was 8.6%, and it came to 49 and 52. So you can see it's like very close by percentage, 49 and 52. So 49 is for the thalassemia, 52 is like very close by percentage in here across all samples. So I think the result is quite reproducible. P progenitors, if we take control versus all patient, you can see the exact peak in here, 8% to in here, like approximately I would say around close to 48%. For cell type annotation comparison, the way it's been done into there, it's like this was the paper which is 8.6%, and if I look at from this analysis, completely different by using the skills that I have developed from there. 8%, 8.6%, it came to like 12%. Yeah, so this one like a 7% in here. Then in here it was around 49.2%, it's 44%, and here was 55%, 45%. So this is very, very close. Like it's a completely different method, annotation, and technology has leaped by this time. So this is a fantastic reproducible rate, and you can see their expression level. You can see these STEM, HSPC, Pro-V cells, late B cells, early B cells, their marker, and you can look at the trajectory, pseudo-time conditions, everything in there. This is very specific in here. CD79, CD79B cell type annotation. This is done part with the C cell transfer as well as cell identifier software that I have developed. You can see the level of expression, IgH, and all those markers clearly given in here. So I think this is quite reproducible. It has also done a small writing to define this, the finding report. And if I am looking in here, major finding of marked expression of CD34 B cells, I have to see how to make it bigger. Okay, that's fine. 8 children with hemoglobinopathies versus control. That's done reproducible. B cell progenitors enriched for lymphoid genes. Done. Next bunch erythroid progenitors. There was not much difference. Trajectory analysis. That early progenitors expanded lymphoid progenitors into the early B progenitors and late B progenitors. Done. B progenitor expansion by flow cytometry, they have done it, which we see in here. Skills selection, how it was done, it's omics flow, it has been used in the back and other things. I think that everything that's has been found into the manuscript has been cross-verified autonomously and done in here. So I think the answer or the question that was asked by Luciano Martellotto and they talked in here probably is that it's not entirely like we are going to need many skills. If you can write if you are an expert machine learning person who can write the proper prompt, and if you are using this skill set from here, you don't need to do anything virtually. Like it works automatically and reproduce the results by using this is a skill. And this answers that AI has been really complementary if you can use it to do the work that takes months within 10 minutes. I hope this helps. Thank